Imagine a rare and relentless form of kidney cancer, one that has left doctors scratching their heads for treatment options. That's the reality for patients with Fumarate hydratase FH-deficient renal cell carcinoma, RCC, a subtype so aggressive and uncommon that clear guidance has been sorely lacking until now. A groundbreaking study from China has shed light on a potential game-changer, the combination of Sintilimab and Axitinib. But here's where it gets controversial. Could this drug duo be the answer? Or are we jumping the gun based on a single non-randomized trial? Let's dive in. In a Phase 2 trial involving 41 patients, this experimental combination achieved a remarkable 56% objective response rate (ORR), with some patients experiencing responses lasting well beyond the study's follow-up period. And this is the part most people miss. The results hint that genetic alterations might predict how well immunotherapy works in this disease, making genetic testing a potential cornerstone of future treatment strategies. For instance, patients with high somatic copy number alterations had significantly shorter progression-free survival, PFS, a finding that could reshape how we approach personalized therapy. The numbers are compelling. A median PFS of 19.8 months and a disease control rate of 73%. But it's not all smooth sailing. While patients with FH germline alterations fared better than those with somatic alterations, the differences weren't statistically significant, leaving room for debate. Is this a breakthrough or a promising starting point? Critics might argue that the study's small size and lack of a comparator group limit its conclusions. But supporters see it as a vital step forward for a neglected patient population. Here's where it gets even more intriguing. In a commentary, experts from Duke University highlighted a previous study where a different regimen, bevacizumab and erlotinib, showed impressive results in a similar patient group. Should we pit these regimens against each other in head-to-head trials? Or is the urgency of this rare cancer's unmet need enough to justify accelerated approval? The FDA's past rejection of Sintilimab for lung cancer, based partly on its evaluation in a Chinese trial, adds another layer of complexity. Are we underestimating the global contributions to medical research, especially in rare cancers? The trial wasn't without its challenges. 13 patients experienced severe side effects, including hypertriglyceridemia, rash, and anemia, requiring dose adjustments or treatment interruptions. Yet, the rarity and genetic complexity of FH-deficient RCC make these findings all the more significant. With no prior prospective trials focused on this subtype, this study fills a critical gap, though it's just the beginning. So here's the big question. Is this the dawn of a new era for FH-deficient RCC treatment? Or do we need more robust, global studies to truly validate these findings? Let's open the floor to you. What's your take? Do these results warrant accelerated approval? Or should we hold out for larger randomized trials? Share your thoughts in the comments below.